Dear friends, this is just a quick overview of the second article that our team is meant to be reviewing. And we just wanted to clarify because there could be some feelings of confusion and overwhelmness in what to do in terms of how to approach the review of this particular study. This study, the effect of physical training on proportion of slow twitch type 1 muscle fibers, which is a novel non-immune mediated mechanism for muscle impairment in polymyositis dermatomyositis, is a follow-on study based on the first article that we reviewed, which was a trial examining exercise resistance and some aerobic exercise in patients with PM and DM by Helen Alexanderson. And if you recall, there were muscle biopsies done in this study, and what this study does, the second article, is to take a closer look at those muscle biopsies from that pilot study. So they're trying to address the physiologic explanation of the results, the improvement that was seen in Article 1. So this is really supportive of the first study, but it's a little scientifically dense. And again, what they're doing is taking a closer look at the before and after muscle biopsies from those research participants. But this time they're also looking at healthy controls. Now, recall that the abstract is our very good friend because it's compact with the most important information of the study, the objectives, methods, results, and the conclusion. And so overall, again, their objective was to look at the composition and the muscle fibers area in these biopsies before and after in healthy controls and in the chronic or the, again, stable or inactive PM-DM patients with that same study. So it's those same patients. And what they did was muscle fiber type and the muscle fiber area were closely examined for changes in terms of appearance and types of proteins that could signal a change in composition. And we'll talk a little bit more about that later. And again, they harken back to the results of the Article 1 study, which where, again, remember, we looked at functional index and the short form 36. There were a couple of other VAS scales as well as a disease impact scale, which we didn't fully understand in Article 1. But the results that they report are that the proportion of, we'll talk about this, type 1 fibers and the type, there's a video I sent explaining type 1 and type 2 fibers and the use of ATP and mitochondria. But the proportion beginning before exercise was lower in these patients that had some chronic residual muscle weakness, even though that they were stable, and that after, and their type 2C was higher, and that was compared to the healthy controls. They controlled for that to make sure that this was a real thing that they were seeing in PM and DM. And after exercise, the percentage of type 1 fibers increased and type 2 fibers decreased. So, and then they go on to talk about the fiber area, that there was a 20% increase in lean muscle fiber area that was also observed. And that usually means an increase in strength. The functional capacity, again, if you remember from Article 1, that that was improved, as well as the physical function in the SF-36 was also significantly improved. And not only that, but they applied another statistical measure to make sure that those really did statistically correlate with each other, that those changes were relating to each other in a symmetrical, in a mirrored way, so that we can really be sure that there was a true association with these changes in the muscle biopsy and in what patients were experiencing in their bodies. So they concluded that this, what we perceive to be a problem in chronic PMDM patients a lot of the time, which is the low muscle endurance, that it could be related to this low proportion of this slow twitch type 1 fiber, which will, again, I might have a chance to explain later, but if you saw the video, you will understand that this is a fiber related to endurance and being able to sustain muscular activity. And that there was a change in fiber type composition, meaning that there was probably a recruitment of those two C's to be transformed to type 1, because they have the power to do that. They have the potential, not the power, to change, to go down a pathway or to become a type 1 more enduring type of fiber versus a fast acting kind of burst type of fiber. And we'll talk about that more. And so, but this improved muscle endurance, and there was decreased muscle fatigue in relationship to this physical training program, which we saw in the Article 1. So again, the introduction that the problem is DMPM patients, as we discussed before, often have long-standing residual muscle weakness, even though their disease does not appear to still be active. And to try to see if there's anything that we could glean from these muscle biopsies to kind of help fill in the so many questions surrounding this phenomenon. And if there's any answers that we can glean by looking at the muscle fiber type composition, meaning the ratio of the type 1 slow twitch and the type 2 fast twitch muscles, and the cross-sectional muscle area. Okay, we will. And something else in this article, there's a more detailed explanation than in Article 1 of the medications. So this could possibly be added to the review for Article 1, and possibly any additional information on the exercise program here. We can probably consider adding that to Article 1. To contribute to the strength of Article 1, or the questions that we had in Article 1. So again, the type 1 fibers, they have more endurance, they have a higher oxidative capacity because they have more mitochondria, more expression of mitochondria, which are the powerhouses of that of energy, which is related to what we call ATP, as you might have seen in the video. And because of this protein called slow myosin, which actually grabs onto some of the fibers and is important in contracting the muscles. So it's a slower process. Whereas in type 2 fibers, they're faster, and they contain a fast myosin, which move very quickly, so it's a fast burst, as opposed to more slow, methodical. Think of it almost like the tortoise and the hare, that old fable You go slow, but you can sustain. But there's something special about these type 2C fibers. So we have, in the type 2 arena, we have type 2A, type 2B, type 2C. But what's special about these type 2C fibers, again, what I mentioned before in the abstract portion of this talk, was that they are considered to be intermediate fibers because they can be more predominantly fast or slow, and can move to develop into the type 1 slow twitch, more enduring fibers, or develop into a real bona fide type 2 fast twitch, oxidative burst type of fibers, using up your energy quick to get the job done. So, but those type 2C fibers, importantly, in normal adult muscle, we don't often see these. We see a composition of the type 2A and B compared to the type 1. So this is a very interesting phenomenon, this type 2C presence in the dermatomyositis and polymyositis with reduced muscle function, which persistent reduced muscle function. So there's a consideration as to whether they exhibit these characteristics, these strange characteristics, could be possibly contributing to what we're seeing here. Is this strange composition related to the fact that there's still residual weakness in patients that we think are really inactive in terms of the inflammatory disease? So, and then scrolling down, right, so basically, again, as you all know, that exercise is considered now to be safe, and it's an important part of recovery. This paragraph here is really just revisiting that fallacy about exercise and myositis. And then they are now talking about the study, Article 1, a recent study that the same authors reported improved endurance with training in a group of patients with inactive but still low muscle function. And they were now we are just talking about the study. So the study demonstrated effectiveness, even though it was not a controlled randomized trial. And there were biopsies that were done, and the biopsy findings that were made it possible to go on to investigate further. So they're really dedicated researchers. They wanted to follow the questions, trying to provide more answers that could potentially explain this phenomena, and also possibly consider examining muscles in such a way when looking at exercise studies in the future. So the first goal of the study was to investigate if the muscle fiber type, meaning type 1, slow twitch, or type 2, fast twitch, composition, and the muscle fiber area, the total area of muscle fiber, the density, the count of muscle fibers in an area, were different in patients with the disease compared to healthy controls. So this is a good comparison because we're double checking to make sure that what we're really seeing is exclusively in people with persistent muscle weakness in inactive DM-PM. So the second goal was to investigate that if that exercise program that was done in the last study on these same patients in those same muscle biopsies related to any changes in the muscle fiber characteristics before and after. So again, we're looking at the types of muscle fibers, type 1, slow, type 2, fast, and also the area. So again, you know what the study is, how patients were enrolled. Again, there was no features histologically consistent with inclusion body myositis. They're ensuring that none of the patients had diabetes or uncontrolled thyroid disease, which could have produced similar results in the muscles. And again, criteria was met. Again, looking at inactive disease, and how did they know that? Because of looking at the muscle biopsies and looking at the MRI results of the quadricep muscles. There was confirmation that the first muscle biopsies that were done in MRIs supported that there was no inflammatory infiltrates or activity, except on one biopsy where there was a perivascular infiltrate without any other suggested findings of inflammation on the biopsy. So again, you know that the tissue was from the vastus lateralis muscle, and you remember that they got it first on one side and then after the study on the other, and that was to decrease any potential artifact. And informed consent was gotten, very important. So the methods. So again, here we have some further information on the exercise regimen that was done in Article 1. And remember, sometimes folks don't report everything because publications are limited to the number of words. And so you're trying to select for the most important things to convey to the readership, which we hope to be able to change somewhat with the guidelines to be more friendly to exercise studies in that regard. So 10 repetitions in several different distant muscle groups with the intention to improve endurance. And again, the exercise program was 15 minutes, 5 days a week, as you remember, and for 12 weeks. And for 5 days a week, there was also a 15-minute walk. Now, a little bit different here in what we had understood in our group, that there was a separate warm-up that was followed by the climbing up and down the stool. That was not part of the warm-up as we had thought in our last team meeting. Exercise for shoulder mobility and grip strength with the pulley. So the question that we had about the pulley apparatus, it was for both shoulder mobility and grip strength, it appears. But remember, we had questions about the way it was written in the Article 1. Strength, we got that information, and we remember that the folks that had the smaller moderate reductions of muscle function, they had a slightly enhanced program with using upper limb weight strength, based strengthening exercises. So again, the assessments in muscle function, you remember the functional index, you remember that there was the SF36. You might recall that there was, but they don't really mention here, that there was a VAS scale for pain and a VAS scale for weakness that the patient was marking in terms of how they felt. And then there was also a disease impact scale that was mentioned in the last study. But again, they're being selective probably because of the limited word count for the article. So going to the muscle biopsies, so what is great to see here that the muscle biopsy reading was blinded for the pathologist who was reading the muscle biopsies. They were blinded to whether they were controls, whether they were pre or post exercise. So this is a strong strength of the study. So they were blinded on all counts, whether it was a healthy control, person with PMDM, whether it was a before or whether it was an after, and they mixed it up in terms of sequence, the samples. So that's really important. Of interest, there was, and this would have been interested to hear about, which I don't think we hear about the central nuclei, but muscles are very efficient, and the nucleus, the generation, headquarters of a cell. In muscle cells, they're often to the periphery because they're thinking about the larger picture of muscle cells in community. So everybody's kind of filling in the gaps and trying to be close enough to other muscle cells to kind of be able to assist, I guess. Whereas in unhealthy muscle cells, the nucleus actually moves to the center of the muscle, and so just trying to keep its own cell alive, really, or functioning well, rather than sharing the load amongst other cells because they're so weak. They've got to just think about themselves. And they looked for inflammatory infiltrates, So they looked at so then going on to examine the muscle fiber type, they did this in several ways, and that is very strong. It was done in several ways to make sure that what they were looking at, what the results that they got, could be validated by another way of looking at that. So the number of type 1 versus type 2 fibers, were they really, did this really exist in the beginning, and did this really change? So how did those numbers hold up if we examine a completely different way of looking at those type 1 and type 2 fibers? So the first way that they did it was looking at ATPase stainings. And so remember that ATP is the energy that's provided. And so I wrote it here so that, so here they tell you what the pH is, and pH at a low value is, a low value is acid, more acid, and at a 10, it's more alkaline. And so what that is telling us here is that, so if they look at it under alkaline conditions with a high pH, that in type 1 slow twitch muscles, it's going to be low. We're not going to, so it's going to be low in type 1 and high in type 2. And then they went ahead and looked at it in an acidic environment with an acidic ATPase staining method. And what we expect to see and quantify in type 1 fibers is that it's going to bring out the type 1 fibers, and we'll be able to count them. And then, but in type 2, it's kind of still high, so it's not so helpful. But what is helpful is looking at it in the alkaline, the differences in the alkaline, and then looking at verifying in with the type 2 that those the absence of, I guess, because we're looking at to see if it's high. And so in this case, it would be the absence is what they found, but more on that later. So very robust in terms of when they did this, the relative number of different fiber types was added up only after viewing around 500 fibers in each muscle tissue section, so in each biopsy section. And then they looked at it in another way. So what they did is, and I don't want anybody to be confused because you're going to see the word antibody. And in here, that's not related to autoimmune disease like PM and DM. This is a technique that we use in lots of different lab tests and in blood and in muscle. And we have antibodies so that we can add them to some blood or attach them to some muscle in this case. And so these antibodies have a special recognition pattern, so that they're going to go and cling to certain things that's the only thing that they're going to cling to. And in this case, the fiber, they're going to cling to fiber type. So for like the type 1 fibers, there's there's the one antibody here called anti-slow myosin heavy chain antibody. So we stain that, or we drop that on the muscle and see how many attach and light up. And then we can also quantify that in type 2 muscles by doing the same thing, staining the muscle and see where it grabs, wash away the rest of it, and then the stuff that remains grabbed onto the muscle, we know is attaching to type 2 fibers, and then we count that up. And so this antibody testing study, we call this immunohisto because it involves antibodies, immunohistochemistry, histo meaning tissue, either blood or muscle, looked at 400 fibers doing this. So that's really robust. And then this part here just talks about the staining results were tested in a regression analysis to make sure that that was real, is my understanding. And then what they did is they looked at the cross-sectional muscle fiber areas of type 1 and type 2 fibers. And so what they did is they looked at 20 fibers, and then they calculated the space that these 20 fibers took up. And so I guess that's a kind of reverse way of doing it, but it's actually probably more precise, I imagine, than blocking out an area of size and then counting in that area. I don't know. I'm not a pathologist, but I'm just making assumptions. But the more muscle fibers in the smaller area tend to be associated with greater muscle strength. So I guess we're looking at density of muscle fiber. And then what they did is, because they wanted to see, because, you know, when muscle that's injured, like in polymyositis or dermatomyositis, there's, in the activity of the disease, there's degenerating fibers. And as the muscle's trying to repair itself, there are regenerating fibers. And the way, again, to identify this can be, again, with this immunohistochemistry, with antibodies that we devised to attach to certain markers that are known to be attached to, in this case, regenerating muscle fibers. And so they looked at that, in some of those types of antibodies that we can wash on and see if they stick are the anti-CD56, anti-vimentin, anti-neonatal myosin heavy chain antibody. And then this talks about the statistical analysis that was done. So the results. They recap from Article 1, the clinical data that we saw, which was improvement. And then we're talking now about the muscle biopsies. So in the fiber type composition, when we're looking at that first way that they did it with ATPase staining, at the beginning, before exercise, this is what they found. We got this from the abstract, right? So now we're just reading more in detail about the results. Type 1 fibers were significantly lower, and proportions of type 2B and type 2C fibers were higher. So that was true in the group itself of DM and PM, and also in the group that were normal compared to the normal group. And so, I might have lied. Type 2A though. The type 2A, which is type 2, which is again the fast twitch ones, the do or die, quick burst of energy, activity. The type A ones though, they did not just differ between the groups. But when we looked at the cross-sectional fiber area, so we see that type 1 and type 2 area did not differ so much. And the male participants, patients, because remember there was limited number of men, it happened to be within the normal values. So so I guess this is possibly speaking to the fact that the area itself did not make too much of a difference. This is where I'm kind of not really able to piece it together too much, and probably need Mariam and Helen's help. So, but the muscle fiber regeneration, so at baseline, there was only a limited number of fibers that expressed those antibodies that signaled this is muscles struggling to regenerate. So what that means is that I guess that we feel that maybe this is a reflection that the disease was not active because there was no active degeneration and regeneration. And then, so looking at the muscle characteristics after the training. So that was before, now this is after. So the fiber type of type 1, the relative proportion was higher, significantly higher, right? And the type 2C fibers were significantly lower compared with before exercise. But the relative proportion of the type 2A and 2B fibers weren't really affected with training. Now, but do you remember what I said about the type 2C fibers, that they were intermediate fibers, that they could really kind of grow up to become type 1 or fully fledged type 2 fibers? So maybe this is what's happening, that they're going down that pathway. So in terms of the cross-sectional area, there was a 25% increase of type 2 cross-sectional area, whereas type 1 cross-sectional area did not increase significantly. And so a fiber type difference was revealed in exercise-induced changes in cross-sectional muscle area. Fiber type times time. So I think this is kind of an inverted approach to saying that the type 2 muscles took up less space cross-sectionally. It's kind of a reverse. I'm getting the hang of this myself. Whereas the type 2 cross-sectional area did not increase significantly. So the markers of regeneration, so it appears that they were unchanged, but the percentage of vimentin positive fibers were. Now the significance of this clinical significance, or actually the biophysical significance of this, I really can't speak to. But it didn't change the other antibody, the neonatal myosin chain antibody. So the CD56 remained unchanged, but there was a change again in this vimentin. So that's interesting. Vimentin is also something that we tend to, well, we'll talk about that another time. So we're going to the discussion. So the authors conclude that the patients were initially found to have lower proportion of type 1, meaning the endurance fibers, and a higher proportion of the intermediate type 2 fibers, type 2 being those burst, muscle strength burst fibers, fast twitch fibers, but these are the ones that can change, remember, in comparison with healthy controls. And then after a 12-week training program, the composition was closer to what we see in healthy controls, so that the type 2 muscle fiber area had increased. Because remember, if I explained this correctly, earlier, and maybe I forgot to explain this, but what they're doing is they're looking at 20 muscle cell fibers and calculating the area. So how much space do those 20 muscle fibers take up? That's my interpretation. So again, there was no changes in the healthy controls. They're supporting their use of ATP ACE use. As having been done in other studies, and also that they again confirmed it with yet another method that was very different from ATP staining, which was the immunohistochemistry to calculate the type 1 and type 2 fibers before and after, right? But even though that there was a low number of patients, they rightly state that the fiber type data changes with exercise were consistent within the group. So even though this wasn't a randomized controlled clinical trial in patients who got exercise and didn't get exercise and look at those comparative muscle biopsies, there was a significant change within the group, and this has strength. And that the low percentage of the oxygen-dependent type 1 fibers in comparison with healthy controls, this is a new finding. That's what they're talking about. These graphs here, they're just really showing us, I think, what is this one? This one is the functional index. So we can see functional index improved in most patients. Each of these dots, each of these lines represents a patient, and that and their scores from beginning to before exercise and after exercise. And here, I think this is the FS36. Yep. So we can see that the functional index from the SF36 in each of these patients that are lined up here, before and after, that most improved. And visually, it's pretty stunning. And then what we're looking at here is we're looking at C, we're looking at the changes in the proportion of type 2 and type 2C fibers. And here, I guess this is an amalgam regression of both types of counting, the ATP ACE staining and the immunohistochemistry. Not sure about that. And then here what we have is the difference, type 1, type 2 fibers. And these are, again, are all the patients looking at the type 2 and type 1 fibers in each of these patients and looking at the before and after here of the area. The fiber area. Okay. So they said, well, could these, what could they be explaining these? Because this was an unexpected finding in the beginning, the type 1 fibers. Could it be because these folks had an autoimmune disease and they weren't moving very much? And they said not likely, because low physical activity is usually also accompanied not just with type 1, but also with the type 2 fibers getting smaller. And that was not seen in the patients. Further, deconditioning, when we look at other types of illnesses like heart failure, where people are sedentary, that we don't see those changes. And then they thought, well, what about glucocorticoids? Because it's true that can cause atrophy. But they affect both types, the fiber type and the composition, but it's known to especially cause type 2B fiber atrophy. So still this type 1 atrophy or lack of presence is still a question. it's not answered by considering glucocorticoids so much. And then looking at age and sex, there was really nothing reported in the literature about that. And actually, this population had more women. Women are known to have more type 1 fibers. So, very strange. So, so they thought, well, maybe it's because type 1 fibers are lower in percentage in DM and PM because they're adapting to the tissue not getting enough oxygen, because we know in these diseases that the blood supply suffers because of damage, number one, and also because of inflammation, number two. So you get a reduced number of blood vessels, and the inflammation can also lead to tissue suffering for lack of oxygen. So this is an interesting, so this remains an interesting question in these patients. So we think, so this is again a seemingly contradictory finding of this percentage of type 1 fibers after training that could possibly have two possible explanations as to why these got better in their eyes. Is that that maybe there was a increase in the aerobic exercise that caused the more oxidative fiber type to increase, and that the other issue would be potentially that that there were several components of exercise, different types of exercises all at once, converging, and therefore really challenging those type 1 muscle fibers to kind of get stronger, get better in the various ways, aerobic and some strength training. Okay, I'm going to skip that. So they conclude that muscle function improved in the physical training corresponding was recorded in proportion to the type 1 fibers and the type 2 fibers. And they find this change to be remarkable. And because a change in fiber type composition after physical training in healthy individuals is not such a, it doesn't often happen. It's a rare thing. So this is a novel thing in these, not just in these patients, but generally in terms of muscle. And that the clinical relevance of this, it's not just we're seeing this in the muscle and so what. This was demonstrated to statistically correlate the improvements that patients described were happening to them, as well as the assessments that were taking place, meaning the functional index and the walk test. So we still need to understand why this is happening. We're not sure why, but it's certainly remarkable. Again, it could be the fact that we're also improving microcirculation, meaning increasing blood vessels, because we know exercise does increase blood vessels, and so therefore increasing the circulation could support the generation and the support of oxidative muscle type 1 fibers. And exercise is known to lower the resistance in the blood vessels, meaning that there's more blood flow so that the muscle no longer is suffering from lack of oxygen. So that could be one. And then finally, they say whether this is true with myositis in other phases of the diseases still needs to be determined. We've seen this in inactive patients with lower muscle strength, persistent low muscle strength. But how does this fare? What kind of changes are we seeing in active disease, in earlier disease? And that we believe that the low muscle endurance that we see in these patients could, at least in part, partially, we have part of the puzzle, maybe not all of it, but we've identified here in this article part of the puzzle as to why low muscle endurance persists in some of these patients, and also that there's a benefit to implementing an exercise program that consists of resistance training and some aerobic training that causes direct changes in the muscle itself in terms of molecular changes and a muscle type, as well as in the real world. So that's it. I hope this helped. It's probably very long, but I do really hope that this made life less burdensome in terms of reviewing this article in comparison to Article 1. Take care, my team and Maya's team. Bye-bye.